Hello all, today we will study the pathophysiology of cellular injury and briefly define cell death. Let's dive into today's question first. A 50-year-old man presents with chest pain and an ST segment elevation myocardial infarction. Histopathology of myocardial tissue 6 hours later shows nuclear chromatin clumping, dilated endoplasmic reticulum, mitochondrial swelling, and loss of glycogen. Which of the following is the most likely classification of the injury? A. Apoptosis, B. Irreversible injury with necrosis, C. Reversible injury with the potential for recovery, D. Reversible injury progressing to apoptosis, E. Sublethal injury with no chance of recovery. If you remember, we have studied cellular adaptations in the previous video, and keeping their mechanisms in mind will help in understanding the pathophysiology of cell injury. Cell injury occurs when cells are subjected to stress that exceeds their ability to adapt or repair. Let's dive into the etiology of cell injury. This stress can be due to number 1, hypoxia, due to ischemia, hypoxemia, and decreased oxygen carrying capacity. 2, inflammation, which is a protective response. 3, physical agents, for example, trauma, temperature extremes, and radiation. 4, chemical agents like poisons, alcohol, and drugs. 5, microbial infections, for example, bacteria and viruses. 6, immunological reactions, for example, autoimmune diseases. 7, genetic defects. And 8, nutritional imbalances with vitamin deficiencies or obesity. Cellular injury begins with hypoxia, which is a deficiency of oxygen. Oxygen is crucial for the process of oxidative phosphorylation in the mitochondria, where it acts as a final electron acceptor in the electron transport chain. This allows for the production of ATP, the energy currency of the cell. Without sufficient oxygen, ATP production is severely impaired and the cell cannot maintain its normal function. As the ATP levels drop, the first major failure is the sodium-potassium ATPase pump, which relies on ATP to maintain a proper balance of sodium and potassium across the cell membrane. When these pumps fail, sodium accumulates inside the cell and potassium leaks out, creating an osmotic imbalance. This leads to water influx, causing cellular swelling. Further complications arise from calcium overload. In a hypoxic state, calcium ion pumps fail, and the calcium accumulates in the cytoplasm. This overload activates destructive enzymes like phospholipases, which damage the cell membrane, proteases that break down proteins, endonucleases that degrade the DNA, and ATPases that consume whatever ATP remains, creating a cycle of damage. Simultaneously, with oxygen lacking, the cell switches to anaerobic glycolysis to generate energy, which leads to buildup of lactic acid. This results in decreased pH, further damaging the proteins and precipitating DNA, impairing cellular function even more. As you can see, hypoxia disrupts critical processes like oxidative phosphorylation and triggers a cascade of cellular events that can lead to irreversible damage. If these changes continue, the cell will eventually die through mechanisms like necrosis or apoptosis. While these are the basics of cellular injury, there are certain changes which are characteristic for each stage of cell injury. These concepts are high yield for any exam. When cells undergo reversible injury, one of the key features is cytosolic swelling. This is a hallmark of early damage and indicates that cell is stressed but not yet irreversibly harmed. Let's break down how this process progresses, starting from outside and moving inwards. First, the plasma membrane starts to show signs of distress, leading to membrane blebbing, small bulges or blisters that form on the outer surface. The microvilli, which are finger-like projections on the cell membrane, begin to lose their structure, further compromising the cell's ability to interact with its environment. Inside the cell, the ribosomes detach from the rough endoplasmic reticulum. This disrupts protein synthesis, further impairing cellular function. At the same time, mitochondria swell, and this mitochondrial swelling results in a decrease in the ATP production. This is a crucial step, as less ATP means that energy-dependent pumps like the sodium-potassium ATPase and calcium pumps fail to work properly. As a result, sodium builds up inside the cell and calcium overload occurs, which exacerbates the damage. The lack of energy also leads to the loss of glycogen, the primary energy reserve within the cell. As a consequence, cells may accumulate fatty deposits in the form of triglycerides. This is known as fatty change, and you may start to see myelin figures, which are aggregates of peroxidized lipids forming within the cells, which are a sign of membrane damage and lipid breakdown. Finally, you may observe nuclear chromatin clumping, where the genetic material becomes irregularly condensed within the nucleus. This indicates that the cell is under significant stress, but it has not yet reached the point of irreversible damage. In summary, reversible injury is characterized by a series of structural and functional changes that, if the underlying cause is removed, can be fully reversed, allowing the cell to recover and return to normal function. Here is an image showing the cellular changes after reversible injury. Moving from outside to inside, you can see the changes in the membrane down to the nucleus where the chromatin is clumping. As cells progress from reversible to irreversible injury, the key hallmark is membrane damage, which marks the point of no return for the cell. Let's explore how this damage unfolds, starting from the outside and moving inward. First, the plasma membrane damage occurs due to the breakdown of phospholipids, which compromises the cell's structural integrity. This leads to the leakage of cytosolic enzymes, which are normally contained within the cell. Their release into the extracellular space signals irreversible damage and loss of function. The damage extends to the lysosomes, the cell's recycling centers. The rupture of lysosomes causes autolysis, where the cell's own enzymes digest its contents, accelerating the destruction of critical cellular structures. At the same time, there is a significant increase in the intracellular calcium. Normally kept at low levels, calcium influx activates the lysosomal enzymes, which further contribute to membrane breakdown, leading to even more widespread damage. Inside the mitochondria, membrane damage leads to increased permeability, which disrupts their structure. This causes vacuolization, or the formation of vacuoles in the mitochondria, and the loss of electron transport chain. As a result, cytochrome C, a protein that is normally contained within the mitochondria, leaks into the cytosol. This triggers a cascade of events that increases apoptosis, or programmed cell death. In the mitochondria, amorphous densities, phospholipid accumulations begin to form, signaling severe mitochondrial dysfunction. Finally, the nucleus undergoes dramatic changes. Pycnosis occurs, where the nuclear chromatin condenses tightly. This is followed by karyohetesis, where the nucleus fragments into pieces. Ultimately, karyolysis takes place, where the nucleus fades as its chromatin is degraded. Together, these changes signify the irreversible injury of the cell, ultimately leading to cell death through necrosis or apoptosis. Once these processes are set in motion, recovery is no longer possible. Once again, use this image to identify the key changes in the cell after irreversible injury. This table gives a comprehensive overview of the previously discussed mechanism. Notice how the membrane and organelles undergo change in each type of injury. These differences determine whether the cell can recover or if it will succumb to death. Moving on to the deleterious outcomes of injury, cell death can take place by two mechanisms. One, a non-inflammatory process called apoptosis, which is a controlled ATP-dependent programmed cell death. Two, inflammatory process called necrosis, which is uncontrolled enzymatic degradation and protein denaturation, resulting in death of a large number of cells. Here is a comprehensive yet consolidated highlight of the key differences between these two. Notice the cause of each of these mechanisms. Apoptosis is a regulated process, whereas necrosis is uncontrolled. Apoptosis is either physiological or pathological and programmed, whereas necrosis is mostly pathological. The cellular changes within apoptosis called cellular shrinkage, chromatin condensation, and formation of apoptotic bodies, whereas necrosis is characterized by swelling, rupture, and inflammation. Apoptosis involves no inflammation as compared to necrosis, which is an inflammatory response. Apoptosis requires energy and is an active process consuming ATP, whereas necrosis is passive and does not require energy. Apoptosis maintains the cell membrane integrity, whereas there is a loss of plasma membrane and leakage of contents in necrosis. Keep these points in mind, and we will discuss apoptosis and necrosis in further detail in the coming video. Coming back to today's question, here we can see all the relevant changes found in reversible cell injury. The findings of nuclear chromatin clumping, dilated endoplasmic reticulum, mitochondrial swelling, and loss of glycogen are characteristic of reversible injury. These changes suggest that the cell is under stress but has not yet reached the point of irreversible damage. If the ischemia is corrected, the cells can recover. So the correct answer is C. Feel free to go over the answer explanation. Here is the key point. I will be covering apoptosis and necrosis in subsequent lectures, so stay tuned for more. Thanks for watching. Don't forget to like, share, and subscribe.